Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research. From advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics, all told as compelling, easy-to-follow stories.
It is so great to be here.
So to start us off today, I want you to think about a sleeper agent in the classic spy movie.
Oh, right, like in The Bourne Identity or The Americans.
Exactly, yeah.
Yep.
You have this character who looks perfectly normal. They live a completely quiet, mundane life, and they just sit totally dormant for years.
They just blend in perfectly with everyone else in the neighborhood.
Right. But the exact moment they receive a specific secret signal, they activate. They go rogue and start causing absolute chaos.
It's a great trope.
It is. But today we're looking at research that asks a strange and honestly slightly terrifying question. What if the cells inside your own skin are acting exactly like those Hollywood sleeper agents?
Yeah, it really is a fascinating framing for what has been, honestly, one of the most stubborn mysteries in oncology.
Because we're talking about how a perfectly healthy-looking patch of skin can suddenly develop a tumor. Right. Months or even years after being exposed to a harmful chemical or some environmental trigger.
Exactly. The danger is already inside the house, just waiting for the phone to ring.
Which is wild to think about. But to understand how cancer literally wakes up, we have to look at what scientists originally believed about where it starts in the first place.
Right, because for a very long time, the prevailing assumption was basically a game of genetic roulette.
Like if you spill a harsh chemical on your skin or get a bad sunburn, any cell could take the hit.
Pretty much. We know environmental mutagens, things like UV radiation or toxic chemicals, damage the DNA in our cells. But tracing the precise cell of origin where that first carcinogenic spark happens has been a massive puzzle.
So where did they think it was happening?
Well, for decades, researchers thought skin tumors might start in the lower bulge of the hair follicle.
The lower bulge. Why there?
That area is really famous in biology. It houses these very well-studied stem cells known as LGR5 positive cells. Alternatively, some people thought the mutations just happened randomly across the flat surface of the skin.
Okay, let's unpack this. If that was the assumption, why didn't the math add up?
Well, they tried to test it, but the older genetic models didn't actually reflect how real-world environmental carcinogens work.
How so?
In the past, scientists used genetically engineered mice where they could manually flip a cancer gene to the on position everywhere in the skin all at once.
That's highly artificial, right?
Extremely artificial. It tells you what a tissue does if you force every single cell to mutate simultaneously.
Yeah, because in the real world, outside of a laboratory, exposure to a chemical or a ray of sunlight only creates rare, scattered mutations.
Exactly. And that brings us to the part that genuinely confused the scientific community.
Okay, what was it?
Recent sequencing studies have shown that perfectly normal, healthy human tissues harbor tons of these oncogenic or cancer-driving mutations.
Wait, really? Just sitting there?
Just sitting there. They are scattered everywhere in our skin, but the vast majority of them never actually form tumors.
Oh, wow. So if you're listening to this and thinking, well, great, I probably have 1,000 tiny sleeper agents in my arm right now, you're probably wondering the exact same thing scientists were.
Which is, if the genetic seeds for cancer are already planted in the tissue, why aren't the weeds growing?
Right. Where are these first initiated mutations actually hiding in the complex architecture of our skin? And what makes them finally act?
So how did scientists figure this out? This is where the story gets really interesting.
I am so ready for this. What do they do?
To answer that, we have to look at a groundbreaking study published in the journal Science in June 2026.
Okay.
The research was led by Dr. Eve Candiba, Dr. Alan Balmain, and their colleagues at the UCSF Helen Diller Family Comprehensive Cancer Center.
And they had some collaborators too, right?
Yes, working alongside collaborators from IRB Barcelona and TwinStrand Biosciences, they decided to perform a completely unbiased bottom-up analysis to find out where these first initiated cells actually live.
And the way they did this involves a technique called fluorescent lineage tracing.
Which is just a brilliant method.
It really is. To visualize this, imagine researchers slapping a microscopic glowing red GPS tracker onto very specific populations of stem cells in mice.
Right. This tracker is a fluorescent protein called TD tomato.
TD tomato.
Yeah, TD tomato. And it makes the cells literally glow red under a microscope.
That is so cool. And it's incredibly powerful because of how that tracker operates, right? It isn't just a dye that washes away.
No, not at all. The researchers genetically modify the target cell so that the instructions to produce that glowing red protein become part of the cell's permanent DNA.
So when the stem cell divides,
It passes the tracker down. Every single future daughter cell that originates from that specific stem cell will also glow red.
you get a permanent visible lineage.
Exactly.
So they essentially color-coded the different neighborhoods of the skin. Like some mice had their lower hair follicle bulge labeled red, others had the surface skin labeled, and others had the upper hair follicle labeled.
That's right. And once the trackers were genetically locked in, they exposed the mice to a known chemical mutagen called DMBA.
Okay, so DMBA acts as the initiator. It causes the initial DNA damage.
Correct. Then they waited a bit and applied a different chemical called TPA.
Now, TPA does not cause mutations, right?
Right. It acts as a tumor promoter. It essentially creates an inflammatory environment that coaxed the mutated cells to grow.
Got it.
Then they simply looked at the resulting tumors to see which ones were glowing red.
And what's fascinating here is that the results totally upended the old assumptions.
Completely. Remember, everyone thought the tumors would come from those famous lower hair follicle stem cells,
Right.
but they almost never came from there. Up to 85% of the fully glowing red tumors originated from stem cells located in the upper hair follicle.
Up to 85%.
Yes. Specifically, these were stem cells expressing the genetic markers LGR6 and LRIG1.
So what about the lower bulge cells?
They contributed to maybe 5% of the tumors?
Oh, wow. So the real culprits were hiding in a completely different neighborhood the whole time.
Exactly.
But wait, I have a question. Why the upper hair follicle? If the whole surface of the mouse's skin is exposed to the chemical mutagen, why do these specific cells go rogue?
That is the million-dollar question.
Because, I mean, if you pour a toxic chemical on tissue, you would expect it to damage whatever it touches. Why did it only take in that one specific microscopic neighborhood?
Well, to figure out the physical mechanism behind that, the UCSF researchers mapped the entire single-cell transcriptome of normal mouse skin.
Okay, let's pause and clarify what a single-cell transcriptome actually is, because that sounds incredibly dense.
Fair enough. It is a bit of a mouthful.
So a genome is a cell's complete library of DNA, right? The total instruction manual.
Right.
But a cell doesn't read the whole manual at once. The transcriptome is essentially a snapshot of which specific pages the cell is currently reading.
That is a perfect way to describe it.
It tells you exactly what proteins and enzymes that individual cell is actively manufacturing at that exact moment.
Exactly. They were reading the factory orders of every single cell, looking for a biological reason why these upper follicle cells were so uniquely susceptible.
And what did they find?
The transcriptome revealed something fascinating. The chemical carcinogen they used, DMBA, isn't actually inherently dangerous when it touches your skin.
Wait, really?
Yeah, it is practically inert.
Meaning the poison isn't poisonous yet. It's like a locked briefcase bomb.
Yes. DMBA only becomes a DNA-damaging poison after it is metabolized or broken down by the body's own cellular machinery.
Oh, I see.
It requires two specific enzymes to convert it into its toxic form. Enzymes called CYP1B1 and EPHX1.
Which are what exactly?
These are cytochrome P450 enzymes, which the body typically uses to detoxify foreign substances. But in this specific case, the enzymes accidentally process the chemical into a highly reactive carcinogen.
Oh, that's a terrible backfire.
It really is. And when the researchers looked at their transcriptome map, they discovered that the stem cells in the upper hair follicle are uniquely packed with high levels of those two specific enzymes.
So the chemical watches over the entire skin, but the upper follicle cells are the only ones holding the exact biochemical keys that accidentally unlock the poison.
Precisely.
Wow, that perfectly explains the where.
Yeah.
But if we go back to our spy movie analogy, that only explains where the sleeper agents are recruited.
Right.
It doesn't explain the dormancy. If the cell mutates, why doesn't it just immediately form a tumor?
To track what happens to the cell after it mutated, the researchers had to use a highly sensitive DNA analysis method called duplexing.
Because standard DNA sequencing is incredibly prone to errors.
Yeah,
right.
Extremely. It's like speed reading a 3 billion word book. You will naturally make typos and read things that aren't there.
So how does duplexing fix that?
It's like having two separate editors independently read the top strand and the bottom strand of the DNA molecule. If both editors don't agree perfectly on a typo, the error is thrown out.
Oh, that's brilliant. That allows researchers to find one true mutated cell hiding among millions of healthy cells.
Exactly. And using that error-corrected sequencing, they proved that a single exposure to the chemical mutagen rapidly induced a very specific mutation in those upper follicle stem cells.
Which mutation?
It's known as the HRAS Q61L mutation.
Okay, and the Q61L part of that refers to a single swapped amino acid at position 61 in the protein, right?
Yes, it essentially jams a cellular gas pedal in the on position, which is a classic driver of cancer.
So the gas pedal is jammed.
The gas pedal is jammed, but remarkably, the car doesn't move.
Wait, what?
Yeah. These initiated glowing red cells with their jammed gas pedals didn't immediately turn into tumors. They persisted in a totally dormant state in completely normal-looking skin for up to 1 year.
A year.
Yes, a full year.
I mean, a year of dormancy doesn't initially sound that terrifying from a human perspective, but we have to scale that to the animal.
Right. A year is roughly half a mouse's entire lifespan.
Exactly. If you translate that biology to humans, we are talking about decades of dormancy. They just sit there doing absolutely nothing.
Nothing. They don't divide uncontrollably. They don't form lumps.
Why does the immune system just sweep them away? I mean, they're mutated.
Because they aren't behaving like cancer yet. To the immune system, they just look like normal, quiescent stem cells. They remain completely dormant until they are exposed to a tumor promoter.
So the alarm clock.
Exactly. In the lab, the researchers use the chemical TPA to promote growth by causing inflammation. But in the real world, outside of a lab, a promoter doesn't have to be a toxic chemical.
What else could it be?
It could simply be the body's natural wound-healing response to a deep scratch, a chronic infection, or long-term tissue inflammation.
Wow. So the mutagen plants the seed, but the promoter is the sunlight and water that forces it to grow.
That is a great analogy.
This means you, listening right now, you could have been exposed to a mutagen a decade ago. The cells mutated, they settled into the upper hair follicles of your tissue, and they've just been sitting there.
Waiting for an environmental alarm clock to go off.
That is wild.
It fundamentally changes how we view the timeline of cancer development. The initiating event and the promoting event can be separated by vast expanses of time.
Yeah.
But the researchers at UCSF uncovered one more layer to this cellular ecosystem, and it answers how these tumors actually physically grow once they wake up.
Okay, lay it on me.
It involves a literal microscopic turf war taking place inside your skin.
A turf war. Because if these cells are sitting dormant for decades, they aren't alone in the tissue, right? The skin is a crowded neighborhood.
Very crowded. And because the duplex-sec technology is so sensitive, the researchers noticed something else while looking at completely normal, unexposed, healthy skin.
What was it?
Even without any chemical mutagens applied, normal skin naturally accumulates spontaneous mutations as we age. As cells divide over time, they make natural typos.
There.
The researchers found widespread spontaneous mutations in a different but related gene called CROSS.
Okay, so in this microscopic neighborhood, we have our chemically induced raw sleeper agents sitting right next to naturally occurring cross mutants.
Exactly.
What happens when the promoter alarm clock goes off and creates that inflammatory environment?
Yeah.
Do they both just form tumors?
You would think so, but no. When the promoter is applied, those natural, spontaneous cross-mutant cells initially wake up.
Okay.
They start to rapidly expand, cloning themselves and trying to take over the territory to form what we call a papilloma, which is a benign wart-like growth.
Right, so they're winning.
But then, a few weeks later, they suddenly vanish from the tissue.
Wait, how does a rapidly expanding cluster of mutated cells just vanish? Are they being physically murdered by the other cells?
Well, it is not a physical stabbing, but it is absolute biological warfare. The chemically induced HRAS mutated cells literally outcompete them.
How do you do that?
Cells in a tissue are constantly competing for limited resources, space, oxygen, nutrients. They also use paracrine signaling, which means they release chemicals into their immediate microenvironment.
Oh, I see where this is going.
Yeah, the HRAS cells alter the environment in a way that actively suppresses the cross clones, starving them out and forcing them to die off.
Because the HRAS cells can take over the space and grow into full-blown carcinomas, which are the invasive cancers.
Exactly.
Here's where it gets really interesting. To prove this was actually a competition and not just a coincidence, the researchers ran a fascinating knockout experiment.
They really did. They took mice that were genetically engineered to completely lack the HRAS gene.
They essentially removed the heavyweight champion from the ring.
Exactly. And when they did the exact same chemical exposure and promotion on those HRAS-free mice, the spontaneous cross tumors survived. They grew and flourished.
which proves the tissue is an actively managed ecosystem.
Yes. The Hross pathway, when activated by the mutagen, doesn't just grow a tumor. It actively suppresses and eliminates the competing cross clones.
It is a literal Darwinian competition for survival, playing out on a microscopic scale.
And the winner is dictated by the genetic background of the cells and the specific environmental exposures they face.
Unbelievable. So if we connect this to the bigger picture, zoom out from the upper hair follicles of mice, how does this change the way we look at human health?
Well, historically, public health has focused heavily on avoiding mutagens, right? Things that actively damage our DNA.
And obviously, avoiding things like UV radiation is still critically important.
Of course. But this research suggests that it's not just about avoiding the initial mutation, it's about deeply understanding the promoters.
Because if our skin and our lungs are already populated with dormant, mutated sleeper cells from natural aging or past exposures, the most dangerous variable isn't necessarily a new mutation.
No, it is whatever rings the alarm clock.
So think about widespread air pollution, specifically the fine particulate matter known as PM2.5.
That's a perfect example.
Some major recent studies have suggested that this type of air pollution might not actually cause new DNA mutations in our lungs.
Instead, that pollution creates chronic, persistent inflammation in the lung tissue. It acts as a powerful promoter.
So it wakes up the dormant sleeper cells that already exist in our lungs from random natural errors.
Exactly. The pollution doesn't plant the seed. It acts as the fertilizer that forces the pre-existing seed to finally grow.
Okay, if cancer is actually the result of this ecological competition between different mutant clones fighting for territory in our tissue, does that change the future of preventative medicine?
Oh, it could completely revolutionize it. Currently, our primary strategy is to wait for a tumor to physically form and then try to kill it with radiation or chemotherapy.
Right.
But if we view the tissue as a competitive ecosystem, future therapies might not just try to poison cancer cells. Instead, we might try to alter the selective constraints of the tissue itself.
You mean intentionally rigging the microscopic turf war?
Yes. If we know the dangerous Hross clones are trying to outcompete the relatively harmless cross clones for space and nutrients, what if a future drug could give the harmless clones an artificial advantage?
Oh, wow. So we could theoretically tilt the ecological balance so that the benign cells naturally outcompete and starve out the dangerous sleeper cells before an invasive tumor ever has a chance to form.
We would be using the body's own natural clonal competition as a cancer prevention tool.
That is mind-blowing. So what does this all mean? Let's bring all of this together and look at the major discoveries we've unpacked today.
Sounds good.
First, environmentally induced skin tumors don't just spark randomly across the tissue. They have a specific origin point in the stem cells of the upper hair follicle, purely because those specific cells happen to contain the unique metabolic enzymes required to accidentally unlock the poison.
Right.
Second, those newly mutated cells act exactly like sleeper agents. They can lie completely dormant, hiding in perfectly healthy tissue for decades until an environmental promoter triggers them.
And finally, your tissue is not a passive canvas where cancer simply grows unchallenged. It is a highly competitive, dynamic ecosystem where different mutant clones are constantly battling for resources and dominance.
We often think of human biology, and particularly cancer, as a broken machine, like a simple mechanical failure where a single genetic switch gets flipped and the machine goes haywire.
Right. But this research proves that our bodies are reactive, shifting landscapes. The ultimate key to controlling cancer isn't just fixing the broken switch. It is understanding how our daily environment interacts with that dormant cellular landscape.
It fundamentally shifts your perspective, right? To realize your own tissue is an active battleground where these epic cellular turf wars are happening right now, without you ever feeling a thing.
It really does.
If our skin and our internal organs are already filled with these dormant sleeper cells, it leaves you wondering what everyday lifestyle factors or common environmental elements might be acting as the alarm clocks we don't even know about yet.
That is the big question.
And if this discovery about how cancer wakes up is just the beginning, what might scientists uncover next?
I can't wait to find out.
Thank you for joining us on this deep dive into the source material. We love dissecting these mysteries with you. Thanks for visiting the Deep Dive Lab, and be sure to follow and subscribe for more fascinating science stories.